we are going to study a detail about the application of RDNA technology. Previously, in the last two parts of presentation, I have showed you, or we have discussed the importance of the RDNA technology, that is the mechanisms and the tools that has been used. Now today we'll be knowing about the application of RDNA technology. Coming to the first application, that is hormone production. It helps in the progress of production of RDNA technology with the help of the bacterial cells like E. coli, for employing them for the fabrication of fine chemicals like stomatostain, insulin, stomatotropin. Coming to the second application, that is transgenic animal production. Now, with the application of RDNA technology, preferred genes can be introduced into an animal, thus to yield the transgenic animal. Now, this technology of RDNA technology helps the animal breeds to search the speed and range of selective breeding in case of animals. It also helps in benefiting the production of healthy farm animals so as to confirm more profit. Now coming to the third application, that is transgenic plant production. Now we all know nowadays with the help of RDNA technology, it is probable to yield a transgenic plant or genetically modified plant. Okay, now numerous transgenic plants have been established with improved properties such as resistance to insect or herbicides or virus, with irrespective or representation of their male sterility, etc. The mechanism to prevent the manufacturing industries or the significant in the help of significant biochemicals or pharmaceutical compounds, etc. Now coming to the fourth application, that is production of antibiotics. Now antibiotics are being manufactured by microbes that are very active against various bacteria or bacterial, viral or protozoan disease. Okay, so few important applications are like important antibiotic are like penicillin, tetracycline, novobiocin, okay, and etc. These are all being searched by RDNA technology. Now coming to the fifth application, that is vaccine production. Now we all know vaccine are a biochemical that are being prepared by comprising a pathogen in reduced or by weakening them or inactivated a state that may be assumed to a human being or an animal to deliberate immunity to an infection, to a specific infection. So now several vaccines have been manufactured biologically via RDNA technology. Coming to the sixth application, that is biosynthesis of interference. Now virus that has been infected or virus infected cells produce very little quantities of interference, which are like glycoprotein in nature. Normally interference have antiviral as well as anti-cancerous property. Like though RDNA technology that helps the genetic factor of human fibroblast, which helps in the yield of interference or interfere or in an human being, which is being inserted or which is being introduced into an bacterial plasmid. These genetically engineered bacteria are then being finally cloned or being cultured so that the gene is able to express and the interference are created, which is fairly high amount. Now coming to the seventh application, that is commercially significant chemical production. Like we all know numerous commercial significant chemicals can be manufactured for more profitable by using the technology of RDNA technology that we have learned from previous. That a few of them are the alcohols and the alcoholic beverages attained through fermentation or by organic acids such as citric acid or acetic acid or etc. and vitamins that are being manufactured by microorganisms. These are the commercially help of RDNA technology that is being used for chemical production. Now coming to the eighth application, that is application in enzyme engineering. So generally this enzyme are encoded by a gene. So there can be variations in a gene, then absolutely the enzyme construction also will alter. So now this enzyme engineering helps the helpers or it is being used for the similar facts and can be clarified as the modification or helps in the modification of an enzyme structure by including alterations in the enzyme which encoded for a specific enzyme. Now coming to the ninth application, that is the prevention and diagnosis of diseases. Now this genetic engineering technique has helped to resolve or help to solve the difficulties of conventional approach for the analysis of disease. It helps in the deliveries or the ways for the inhibition of several diseases such as AIDS, cholera, etc. Now, with the help of this monoclonal antibodies that are being useful, that are being used as a tool for this disease diagnosis, are being produced by utilizing this technique you call hybridoma technology. Now, this monoclonal antibody, that the process is that this monoclonal antibody bears a specificity to a particular antigen. These are used in the analysis of disease for owning their specificity. This genetic engineering helps in the fabrication or helps in the permission of fabrication of this hybridoma, which is a cell attained by the fusion of a lymphocyte cell which is capable of producing an antibody and a tumor cell. The fusion occurs between a tumor cell and the lymphocyte cell. Now coming to the last and the foremost application, that is gene therapy. Now this gene therapy is very favorable to the areas where the genetic engineering for human being. It is very favorable for the genetic engineering of a human being. Now it includes like the delivery of a particular gene into a human body for the correction of the disease. So it is the treatment of a disease by transfer and by the expression of gene into the patient's cells so as to confirm the resorption of a normal cellular activity. So the gene therapy may be categorized as a somatic gene therapy and a germ-line gene therapy. Now, these are the applications of RDNA technology. These are a detailed, I have told you a detailed study or I have told you a brief study on these applications. Study the application or we'll discuss the application of genetic engineering in medicine. Now we can first see the first application is DNA vaccine. So I have created an image. You can see just beside the DNA technology. You can see a symmetric diagram or a diagrammatic view that I have given you, written as preparation and administration of DNA vaccine. So here I will tell you what is the mechanism of the DNA vaccine. First, it includes the direct injection of plasmid DNA that has been encoded an antigen protein which is then participated or articulated inside a cell of an organism, thus creating the organism's efficiency disease resistant. Now the vaccines are vaccinated through needles or through the skin. Though less quantity of cells are affected, the reaction, which is not fully unstated, somehow efficiently immunize the body. Coming to the second application, that is gene therapy. Now this gene therapy is a cloned functional copy of gene that are being introduced in an individual to help in balancing the individual defective copy. Now this gene therapy is employed for a number of diseases comprising such as SCID as well as hemophilic. Now coming to the third application, that is manufacturing of pharmaceutical products. Now you can see a diagram, or I have given a table, like the first therapeutic product, that is vaccine, and its application is being used in utilization and antigen. Now coming to the second therapeutic product, that is insulin. Its application is that it helps in the diabetic treatment. Now the third is alpha 1 antitrypsin, that is AAT. It is help in the treatment of AAT deficiency. The fourth is IL-2. It is help in the treatment of cancer, immune deficiency and HIV. Fifth is the tumor necrosis factor, that is for treating the tumor cells. And the last is interferon, that is been helped for the cancer treatment. These are the applications of genetic engineering in medicine. We are going to study the combined application of a rDNA technology and a genetic engineering for the production of various substances. Now the first one that we are going to discuss is about the fabrication of interferon. So how does it work? Now first we have to know what is interferon. So interferon are the natural glycoprotein that have been formed by virus infected eukaryotic cells, which protect host cells from virus infection. Now these substances are named as interferons as because it interfered with the intracellular development of virus. So these interferons are manufactured by living animal cells, both in vivo and the cultured cells. Now the substance in an virus that is responsible for interferon synthesis by the host cell is known as a interferon inducer. Now it is normally a double-stranded RNA. The activity appears to exist in an polyribonucleotide, which are higher in helical content. Now for a double-stranded RNA virus such as reovirus that can act as an interferon inducer deprived of replication. Now a single-stranded RNA virus can act as an inducer only after the replication, when they form a double-stranded replicative intermediates. Now here you can see the DNA virus can also prompt interference, apparently owing to a overlapping transcription of viral DNA as detected in the case that we can see the diagram over here. Now the interferon molecules that are leaving the producing cells reach the neighboring uninfected host cells that are related with the cell membranes or the nuclear membrane receptors of the cells. Thus the cells are brought to synthesize the antiviral proteins. These antiviral proteins are the actual agent that helps in the delivery or delivers safety to the host cells against the viral infection. The second application, that is fabrication of hepatitis B vaccine. Now we can see there are several stages that are involved in this process. Coming to the first step is that isolation of HBs antigen, fabricating gene from the HB virus by standard isolation technique. Now the second that we can see in the diagram is a plasmid DNA that has been taken out from a bacterium E. coli and then is cut with an restriction enzyme, that is the E. coli making the plasmid vector. Now coming to the third point, that is insertion and then location of the isolation of HSB antigen that are used for constructing gene that is being into the bacterial plasmid vector, which helps in the establishment of the recombinant DNA. Coming to the fourth is that the recombinant DNA of the comprising the target gene is hosted inside a yeast cell. Okay, now that is creating a recombinant yeast cell. The fifth is this recombinant yeast cell helps in proliferation in the fermentation tank and which helps in further the yield of HBs antigen. Coming to the sixth, after 2 days we can see the yeast cells are cracked, and then they free this HBs antigen. Now this fusion is treated for the help of extraction and HSB antigen are further being purified. Now this HBs antigen are joined with preserving and agent and extra constituent are bottled. Okay, now this vaccine is now ready for the injection in the human. This is the full mechanism that I have told you with the help of this diagram. If you can remember this diagram with all its the ones that are written with its details, you'll be able to understand. So just follow this diagram very nicely, you'll understand the full mechanism. Last application, that is fabrication of insulin. So in like the hepatitis B vaccine, here also in the fabrication of insulin there are some steps. There are 6 steps that have been involved. So going to the steps, the first step that has been involved in this genetic engineering is that human insulin is removed from the pancreas cell and then this insulin is being producing gene is been isolated, and then further a plasmid DNA is extracted from a bacteria and cut with an restriction enzyme and further help in developing a plasmid vector. Now the third is this human insulin producing gene is further being inserted into a bacterial plasmid vector, which helps in the formation of rDNA of a human insulin producing gene. Now this rDNA is further introduced into a bacterial cell to form the recombinant bacterium. Now this recombinant bacteria is further reproduced in a formation tank and helps in yield of human insulin. Now this insulin is being extracted and then been purified and then been bottled, and this is the full process of the fabrication of insulin, and further it is ready for injection in any diabetic patient. So this is all about the application of fabrication of insulin. Now all in this presentation I have told you about the applications of RDNA technology, genetic engineering, and both RDNA technology and genetic engineering. Next slide I'll be telling you about the PCR technology.